Hello, everyone, and thank you for joining today's UMB ICTR Enrichment Seminar. Before we get started, just a reminder that this session is recorded and will be posted to the ICTR website in our education section for your future reference. We plan to reserve the last 10 to 15 minutes for questions and ask that you please post them in the chat or feel free to use the Zoom Q&A feature. Finally, as you leave this session, we will redirect you to a survey. We value your input for future presentations, so please take a moment and share your thoughts with us. And now, I am so pleased to introduce our guest speaker, Dr. Maureen Kane from our School of Pharmacy. And just a quick bit of information, Dr. Kane joined the School of Pharmacy as an assistant professor in 2009 and is now a professor of pharmaceutical sciences and the director of the Mass Spectrometry Center. Dr. Kane is well known to the ICTR as she is also a key leader in our UMB ICTR Drug Discovery and Development Core. The Mass Spectrometry Center is an organized research center on our campus with an emphasis on collaborative research and the acceleration of drug discovery and development. Dr. Kane will highlight key capabilities and will also include how investigators can access this resource through the ICTR. I will periodically add that link to our ICTR website for drug discovery and development resources. And if you have any questions, just add those again to the chat or to the Q&A feature in Zoom. And now, let's welcome Dr. Kane.
Great. Thanks, Wanda. I'm very pleased to be here today and share with everybody some of our exciting capabilities that you really do have a lot of great access to on this campus. Whoops. See how I control my slides? There we go. So Wanda just gave us a nice intro about the goals of our center. Just to emphasize, we work collaboratively for the most part with people because we find that if we work with you as part of your team, it produces better quality research. We understand your goals. We kind of discuss with you what the analytical options are. We help you with study design, analysis goals, and in that way we can produce the highest quality science so we can publish novel research and obtain sustainable funding. We also are a training ground for people to not only get hands-on training, but to interact with us and get a good knowledge base of how to implement these types of techniques. And so I put this slide in here. I won't spend a lot of time on it, but this is some of our key metrics. We're a multi-million-dollar facility. We have a variety of mass spectrometers of a variety of function, everything from omics to targeted. And we've supported research at UMB over the past 5 years to over $110 million in total funding that's come into UMB. So we've been integral into the research enterprise here at UMB for various types of questions. And you can review this later. Well, I think we're going to share the slide deck. And so as part of the ICTR, this is a great resource. If you go to the ICTR webpage and you look at the investigator resources and go to the drug development and the discovery and development core, you'll find a link to the Mass Spectrometry Center where it talks about what kind of resources you could get. And I just wanted to highlight this here because there's a great opportunity for you to get some basically subsidized resources through the ICTR. And this is just some examples here of what types of kind of things we give you consultation. ICTR essentially provides some funds to be able to do some small types of projects, to be able to get pilot data or test feasibility and be able to support grant applications so that then we can go after that sustainable funding. And there's lots of information that's on the webpage. I think Wanda's going to post the link, and I encourage you to go there and set up a consultation if you want to talk more about how to access these ICTR resources. So our goals for today are really to give you an overview of the types of questions you can ask using mass spectrometry-based analyses and highlight some of the various capabilities of the center and also highlight some of our kind of ongoing collaborations and pubs. And they're throughout the presentation that you can kind of look at later if you see an area that you're interested in. And so I've kind of divided this up into a couple different areas today. And like I said, I'm not going to go into too in depth on any of the particular things. I want to highlight more the diversity of different tools that you could implement in your studies by working through the Mass Spectrometry Center and highlight those types of things. And I broke this up into things that are sort of drug discovery related, omics type studies, and other types of analyses that help you understand either your disease, your drug target, identify your drug target, develop biomarkers, lots of different types of questions that aid in drug discovery. And then drug development, we have a whole suite of tools that, like, if you have a target and you want to further characterize that target, or you have a drug you're already working with and you want to understand, properties of that drug, how the drug works, PK/PD, things like that. We have a variety of tools to support that too. So I'll talk about each of these different types of approaches. And so I'm going to first talk about one of our probably most popular resources, accessed through ICTR and also through a variety of people on campus. And these are our suite of proteomics techniques that have a lot of versatility in the types of questions you can ask. And so this slide has a lot on it, but it kind of highlights that there's kind of 5 main areas of different types of proteomic studies that we kind of commonly employ. And these are very applicable to a wide variety of biological samples. It could be tissues, it could be cells, it could be fluids, it could be isolated subcellular fractions, a variety of things. We employ a variety of enrichment techniques depending on your goals. And so just to break this down, I'm going to go through some examples of each one of these things. These are kind of the main areas of proteomics. And so the first one I'll talk about is global proteomics, which is kind of a discovery approach. So this is an unbiased profiling of your entire proteome. So it's a priori, no hypothesis needed, and it has a pretty good dynamic range if you want to explore your questions. And so types of things that you can do with this type of approach is you can basically do comparative analysis, kind of A versus B, my control versus my disease, my drug treated versus the control. And again, you can do this in tissues, you can do it in fluids, cell culture if you have a cell culture model, or if you have sorted cells. If you have subcellular fractions, if you do a pull down, you can enrich for various things. We've done a bit of exosome analysis lately. So pretty much most types of samples are amenable to this global proteomics. And then paired with the global proteomics analysis, so basically you're assaying all the proteins that are in your system. And then we pair this with bioinformatics analysis, where we often do pathway analysis, comparative network analysis, upstream regulator analysis, basically things like that, as well as a variety of gene ontology analysis to relate things back to their biological processes or cellular functions. And this can help you identify, key dysfunction in your system. It can help be hypothesis generating. It can help understand mechanisms. So it's really a versatile set of experiments. And so I'm just going to highlight some of the things we've been working on kind of lately. One of the collaborators who we've done a lot of work with is Marta Lipinski over in School of Medicine. And this is a highlight of some global proteomics. And this was actually on an isolated subcellular fraction on lysosomes. And so from this model, the organelle was isolated, and then we performed proteomics. And in this case, it's a neurotrauma kind of aging study. And so this is just the aging data, and it shows that, like, longitudinally, we assayed the proteome profile to look at what was different as a function of age. And then, the follow-on part of that study is after injury, is the aging process altered? And so proteomics has been a key part of this study in identifying mechanisms. And so this is a nice highlight of that. And there's a variety of papers we have with her lab. This is another global proteomic study I wanted to highlight, because this one shows you can analyze a diversity of samples. So in addition to things like tissues or cells or fluids, you can also do kind of more unique types of samples. And in this case, we worked with Ryan Pearson in School of Pharmacy, who does a lot of nanoparticle-based things, and he was interested in what proteins were associated with the outside of those nanoparticles. And so we used this proteomics approach to profile what was the protein corona of these nanoparticles under a variety of conditions. And so this had to do with inflammatory diseases, and we took this global proteomics, and we were able to characterize that protein corona and then be able to relate that back to his study. So this is a very versatile technique in terms of the types of questions you can ask. And I wanted to highlight one other study on this, and because this is an example also of a kind of inflammatory study. It's from Prajwal's lab over in School of Medicine, and it's a brain injury study where he's studying a hemorrhage, and we're analyzing CSF. And the goal of this study was to really look at the inflammatory proteome, which tends to be of lower abundance. And so in this kind of global proteome example, we did an enrichment. And so we've done a number of kind of experiments to show that if we use nanoparticles to enrich the CSF, it enriches these lower abundance inflammatory molecules, and you get much better coverage of those. And so in this case, where they're very interested in what's going on with inflammation, we were able to enrich, get much better coverage, and then be able to relate this back to specific biological processes to help them understand basically their injury model. And so this has been a really nice development. We're preparing a manuscript on this, but this is an example of how you can do various strategies to favor a subset or enrich to get better coverage of a specific group of proteins that you might be interested in. So the last kind of global proteomics thing I wanted to highlight is while we work a lot of times in human or mammalian animal models, we also have done a lot of stuff with bacterial proteomics. So if you study other kinds of things, this is also a possibility with this type of approach. And so this is just an example from Angela Wilkes' lab, who's a longtime collaborator at School of Pharmacy. She studies Pseudomonas and mechanisms to develop new antibiotics. And so we've really used global proteomics widely of the bacteria to understand various targets within them. And in this case, we not only did the global proteomics, but used a variety of network analyses to identify signaling nodes, which were affected by, in this case, mutations to a key protein in heme metabolism. And so I'm going to talk later about metabolomics, but one of the things that I'd encourage people to think about when they think about using the Mass Spectrometry Center is how to maybe attack their question from different directions. And in this case, with Angela's project, we not only did the proteomics, but we did companion metabolomics. And in this case, I've kind of highlighted at the top, these were some enzymes that were identified from our global proteomics, and it's laid out in a pathway where all the ones labeled in red are the enzymes. And then we designed a targeted metabolomics panel to assay the actual metabolites that are getting converted by all these variety of enzymes so that we could support the changes in those enzymes resulting in changes in the biomolecules that they regulate. And so integration of both approaches made a much stronger story. And I encourage people to think about framing their questions with such kinds of things if it's appropriate. Okay, I'm going to go on to the next type of proteomics. This is a targeted proteomics. And so the difference between this and what I just talked about is that in the global, it's untargeted. We're assaying whatever is there. In a targeted proteomics assay, you're selecting specific targets and you're only assaying those. And this often will give you much better quantitation. And in the example I'm showing you, some of the protein questions that we ask require better quantitation, like determining a protein half-life. And in this case, this was a protein my lab was studying where there's mutations in humans. And when we did the in silico modeling, they were predicted to be destabilizing. And so we wanted to pair this with actual experiments to understand the stability of those proteins. And so we developed this targeted proteomics approach. We did stable isotope labeling to be able to determine the half-life. And then we were able to measure both the wild type and the various mutant half-lives, and we were able to support that shorter half-life in the destabilized proteins. So taking a targeted proteomics approach sometimes can be very useful when you want to answer questions that have more of a nature like determining a half-life or something like that. Another popular proteomics approach that we often employ is PTM analysis. So post-translational modifications come in a variety of flavors. Phosphorylation is probably the most widely one we've used, but they also include other types of things like glycosylation, histone modifications, methylation, etc. So I'm just going to show you a couple examples from that. But basically, for things like phosphorylation, it's a low incidence kind of modification, you enrich for it, and then you can assay that phosphoproteome. And so we have well-developed methodologies to do that enrichment. We also have enrichment methods for glyco, and then you're able to basically identify where those modifications are and determine their stoichiometry. And so here's just an example from a phospho analysis we did with a collaborator. And in this case, it was a survey of the phosphoproteome to understand, what was modulated in this particular situation. And so you can take kind of an unbiased approach to a phospho study where you can just assay, what different proteins are phosphorylated, and then from that study you also get information on where they're phosphorylated. And or you can take an approach where you really look more specifically at your say your favorite target or your protein of interest or your specific pathway. In this case, we took a broader view and characterized the whole phosphoproteome. And so one other thing you can do with PTM analysis is say you have a drug molecule that covalently modifies a protein. It's very much like a PTM analysis. It is a PTM analysis essentially. And so this is a example highlighted from Paul Shapiro that we worked with. And Stephen Fletcher in our department has synthesized a series of molecules which basically covalently bind to ERK, and they modulate the function outside by binding to ERK outside the active site. And so it was really important and for this project to show where it was binding. And so we used a mass spectrometry based approach to be able to show where that covalent modifier, which residue, the drug molecule had covalently modified. And then as they've made subsequent analogs to this in new generation compounds, we've done a variety of additional analyses just like this, where we've taken the proteomics approach to be able to identify where those molecules are modifying the protein. So not only for on-target, residues that are modified, but also to survey to see if there are any off-target effects. And then we've also subsequently worked with Paul to study, global and phosphoproteomics approaches to understand that modulation of the ERK protein. So you can take kind of a multi-technique approach to your questions if you're interested in a variety of pieces of information. So the next type of proteomics I'm going to talk about is chemical proteomics. And so this is kind of a more of a custom type scenario where there's different types of questions. So in the stuff I've talked about so far, you're kind of comparing, A versus B or drug treatments. In this case, these are a little bit different style approaches. And so the first one I'm going to talk about is thermal proteome profiling. And we use this one with Vincent Najjar to identify a drug target. It basically works like SETSA, where, you treat you have your cell lysate or your isolate protein, and you treat it with the drug, and you basically subject it to a temperature course, and the binding of your drug to the protein target will stabilize or destabilize it. And then you can assay the proteome, and as a function of temperature, those ones that are stabilized by the drug binding will be able to be distinguished from the untreated, and those identify your drug target. And so we did this very nicely with a study with Vincent, and he was able to validate those targets. And so this was really, really helpful. And we've also done this with Angela Wilkes' lab to identify targets of novel proteins. And so this is a really useful approach. If you're not sure of your target or you think there might be multiple targets, it's a biophysical technique which utilizes some basic proteomic approaches that can help identify your drug targets. So another thing that you might be interested in when you're looking at specific targets is who they're interacting with. And so things like BioID or TurboID, where you have basically an enzyme fused to a protein, your protein of interest, and anything nearby will be covalently, basically labeled, and then you can isolate those labeled proteins and analyze what they are. And so we've used this with people lately. This is really useful to basically understand who's in the vicinity, who's interacting with your target protein. And so if you want to understand that, you can take an approach like this. And so this is a modification to the proteins that are nearby. And so it's a chemical labeling where you can then get information on who's in the proximity to the bait. Um, And then one other one I wanted to highlight for chemical proteomics is one we've done recently with Mark Samard's lab, where they labeled, they used this direct detection of biotin containing tags approach, where basically the you buy you, the proteins get biotin tagged, and they were able to look at the transport of those proteins through the spinal cord axons. So this was really cool approach that takes a little bit different, methodology to the other types of labeling, but it allowed us to give them information on the transportome in these spinal cord axons. And so this was a really cool approach. It tells, the proteins that basically were transported and basically we've shown, what they're associated with. And then it helped them to understand this a bit better. So this is a new approach we've kind of employed. Um, and this is an exciting approach if you're answering or you're asking questions like this. We may be able to help you with something like this. And so the last thing I'm going to talk about is spatial type resources. So sometimes where your protein's located is important, and we have a variety of kind of approaches you could take, everything from just physical dissection to laser capture microdissection to sorted or single cell kind of type analyses. This is great for samples like tumors or lesions that are heterogeneous, like wounds or 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 burns, and also tissue regions if you have. A tissue that has specific regions that have unique functions. This can also be kind of useful. And so you can do physical dissection. This is an example one where it was a burn, but it was a pretty big scale. So the samples are able to be physically dissected, and then you can take just that global proteomic approach. And in that case, it's pretty straightforward. But what we've added recently is we've added laser capture microdissection. And so this really allows us to microdissect specific regions and tissues. I think we're down maybe in the 5,000 or less cells kind of range at this point in time. We're still kind of optimizing this a little bit, but we're using laser capture. And so like you could take regions from, say, your tumor, or you could take them from your injured region, and you could survey specific regions according to histology or to whatever marker you're using in your tissue, and then we can perform the proteomic analysis of those micro dissected regions. And so this just shows you kind of we've done a systematic test of the amounts and kind of the coverage we get, and it's a very reproducible workflow that's able to give you quantitation. So this is low input. We're not quite to true single cell at this point in time, but we're down into very low input at this point in time. So this pairs nicely with those other types of spatial workflows. And so this is a good transition into spatial. And so one of the other tools that we have in the Mass Spectrometry Center is mass spectrometry imaging. And so I'll talk about that now. And so this is like a molecular histology. So we have two different modalities that we have at the Mass Spectrometry Center, matrix-assisted laser desorption, MALDI, or desorption electrospray, DESI. And basically this technique combines mass spectrometry with histology-like information. You don't need any labels like you do with, antibody-based, kind of staining. And you're able to spatially localize molecules and, some of the main questions that we use this for, either to characterize the disease or you do things like ADME studies where we will localize a drug. And so this is really amenable to drug molecules, metabolites, degradation project, biomarkers, all kinds of different things. And so the cartoon here just shows you, MALDI uses a matrix, DESI does not. No, and so DESI is kind of nice because you can do histology on the exact same slice that you did the mass spectrometry on, where MALDI, we usually take a serial slice. And then you can display the ion from your mass spectrum for a spatial localization. And so I'm going to highlight a few examples of these because I think these are, things that can really add to people's studies. And so in this one, this is a collaborator we had over at Hopkins, and they were looking at paired tumors and lymph nodes. And we had assayed a series of metabolites in a couple different pathways that they were interested in. And we were able to show here, you can see that the localization of the various metabolites varies in the tumor and in the lymph node. The little one is the tumor, the big one's the lymph node. And then we linked these through kind of like their connections through pathways in the middle. It's kind of a busy slide, but it just shows you that like you can really assay a variety of different molecules. You can do pathway and in system network analysis, and you can look at variety of metabolism indicators. And so one of the other things we did in that study is we did traditional kind of IHC to identify certain cell populations, and then we overlaid that with the MALDI. And so in this case, where, if traditional H&E kind of distinguishes your tissue regions, that's great, but sometimes you need an additional marker. And so in this case, we used IHC to mark for specific types of immune cells, and then we overlaid that with the MALDI to localize the basically metabolite signature of those specific immune cells. And so that was a pretty productive approach that if you're thinking about trying to understand, maybe tumor heterogeneity or some other kind of tissue heterogeneity, you could use this type of approach to answer those types of questions. Oops. This is another example of using mass spectrometry and imaging. This was another collaborator over at Hopkins for interested in endocrine disruptors and how they collected in skin and how they affected some disease pathology. And so this is just kind of highlighting some of these things. These are from a recent paper. And the top shows bisphenols, the bottom's phthalates. And why I wanted to show this is because this gives a great example of how to pair this and not only tissue. So these were tissue samples from human. And then we paired these with ex vivo cultured cells. And so the bottom one, we imaged the cultured cells to understand in that in vitro model which ones were kind of getting taken up into the cells. And so this is an example of one way you can employ mass spectrometry imaging. And then the last imaging one I want to highlight is some work we've done. I talked a little bit about our collaboration with Marta Lipinski earlier, but we've done a lot of imaging with her about lipids after traumatic brain injury and their localization to sites of injury in the brain. And I wanted to highlight this is because we did actually quite a bit of new technology development in this space, because one of the challenges when you're imaging lipids is that you can have lipids that are different lipids, but they have the same mass. And so we basically developed some, we got an instrument and we developed some methodology where we employ ion mobility to separate those ions that have the same mass in the gas phase and then be able to tell them differently. And so the example I show you here are two lipids that have basically the same mass out to like two decimal places. And then the peaks that you see in the mobilogram just show you can separate those. And if you then look at the spectra, basically distribution of those in the images, you'll see that those two lipids are very differently distributed. And so that if you hadn't separated those, it would be very difficult to see the changes in those individual species. They would have been lumped together. But with the power to be able to distinguish these isobaric lipids, now we get a better understanding of the biology and the spatial distribution of these lipids. And you can see in the top right of the brain image, that's where the injury site is. And you can see there's quite a localization of the one lipid that would have been missed if those were lumped together. And so we've employed this in MARTA studies basically to show the lipid dysregulation at the site of injury. Again, in all these images, the injury site in the controlled cortical impact model is in the top right of the brain. And we did this to show, collection of lipids at the injury site. And we also did a longitudinal study where we looked at this as a function of aging. It's kind of interesting. The one I showed you, this is a ceramide, and you can see that at 3 days, that one very brightly, the second one in is shown. And then over time, there's still dysregulation in this lipid, but it's not, it's in sites that are actually not the immediate site of injury. So this is an ongoing study. There's a paper that's in review, and there's more studies that are in preparation right now to come out from this work. And then one of the other things that we've done is we've taken a multi-omics approach in those studies, and they've combined proteomics and lipidomics and imaging. And this is just an example to show how you compare some of those things. From LCMS-based data, there's just an example here of how a generic kind of lipidomic separation, and we quantified stuff from this model of MARTAs, and then the graph of the lipid species, you can see that were changed in the TBI. We then localized those to the images, and you can see that the images agree with the LCMS data. So you're able to assay those lipids and then show their spatial distribution within the tissue slice to show a molecular histology of that. And so I want to move on now to talk about metabolomics. And metabolomics is covering kind of both metabolites and lipids. And metabolomics has some unique features different from the proteomics, so that it's often very sensitive to disease and drug exposure, and very good for biomarkers, in easily translatable across species. And so one of the so we take two different approaches with the metabolomics. One, we have a very large targeted metabolomics kind of discovery approach that I'm showing you here. And then we have very specific targeted custom assays for a lot of things. And we can also make new ones for you depending on your question. And so from the targeted metabolomics discovery thing, we use the commercial Biocrities kit. I've used this in my own studies for probably nearly 10 years now. It's extremely reproducible. And the high points of this approach, as opposed to an untargeted metabolomics, is all our metabolites and lipids on this panel are identified. And right now, they've expanded it to the point where there's, I have the number of markers down at the bottom, but the ones we've been using now is the 500 and the 1000. The 1000 just came out. And there's really like 1500 to 1800 metabolites and lipids on this panel. It has really good coverage of things that are biologically relevant to a lot of different situations, and everything's identified. It's all quantitative. It's very reproducible, kit to kit, and over time, year to year. So this has been a great metabolomics approach, and I'm just going to highlight one of the things. So we use this in our own work for biomarker work for development of biomarkers for radiation-induced injury that is used in the drug development of countermeasures. And we've used a multi-omic approach using proteomics, imaging, and metabolomics. And shown here, you can see there's multiple papers we published with this targeted metabolomics approach for biomarker discovery. And I'll just show you a couple examples of this, but I wanted to highlight that when our tools that we have, you can go right from the beginning to the end of really biomarker discovery through validation. So having a good defined model is important, but you can do biomarker discovery with us. Then we can help you identify and characterize and validate all of those biomarker species. So it's such that you define that biomarker clinical endpoint relationship. And so I'll just show you an example of that. This is just some examples of the data from some of our radiation biomarker discovery work. And so the heat map just shows one of the time points. And from that time point, this was one time point from a longitudinal study. And then I highlighted a couple different things because from the longitudinal signature of those ones that are kind of in red, we looked at their basically metabolite level as a function of time, and we correlated these with the survival. And so one of the things you want to understand with your biomarker is how does it relate to your clinical endpoints. And so from this quantitative metabolomics data that we generated, we're able to do things like correlate with clinical outcomes like survival. We also correlated it with histological scoring. So that quantitative data you get from the metabolomics is very rigorous, it's very reproducible, and it's able to be correlated with these outcomes like survival, histological scoring. In this case, it was a measure of GI injury. We had a very high correlation with this. This is a very good biomarker candidate. And then the other area that metabolomic biomarkers have been very useful is assessing drug efficacy and toxicity. And so this is an example of assessing basically drug efficacy and biomarkers that are um, responsive to the therapeutic. So this is also a radiation study where we had basically unirradiated, irradiated, then treated with two different doses of a drug. And the goal was to identify basically biomarkers that had pharmacodynamic potential, meaning they were drug responsive in a dose dependent manner. And so we were able to use this targeted metabolomics approach. And I showed you just one example where you can see that over time we had a metabolite basically time course, then we had two different concentrations, and you can see that we have concentration dependent change in the metabolite back towards baseline with the treatment of the drug. And so this helps identify biomarkers that could be used in the basically development of the drug is a surrogate endpoint. Okay, and so the last thing I want to kind of talk about, and I want to leave some time for questions, is some of the tools we have in the quantitative LCMS area. And this is things like drug quantification, preclinical screening assays, PK/PD. And so most of these types of experiments rely on a targeted metabolomics that's a custom quantitative LCMS. So it's absolute quantitation targeted to your specific molecules. And this is very versatile as well. It could be for endogenous molecules, biomarker candidates. You can use it to validate your omics hits. You can quantify drugs in patient plasma. You can use this to do PK studies in animals and people. You can use it to assess kind of these drug candidates and their characteristics. Lots of different questions you can ask. And so we do everything from develop the assays, validate them, transfer stuff from the literature, verify they work, and then use those to answer the questions. And so just a couple examples. This is just an example of a drug quantitation and drug analysis. And in this example, this is with Jim Polly in my department. We developed an assay for the drug molecule and one of the major metabolites. We validated that, we published that method, and then we used that method in his study where he was looking at PK of various formulations and the effects on the drug disposition based on the formulation. And so then we use this rigorous quantitative assay to be able to do those patient PK profiles. So, kind of we can do everything from beginning all through the PK analysis. And then one other thing that we've kind of added recently is some of these preclinical assays. And so this is just a couple examples using some metallo drugs. These were synthesized in Feixian Shu's lab in my department, and Angela Wilkes part of this project as well. These were metallo drugs where we were interested in understanding their properties like KD solubility, microsome stability, permeability, things like that. And so just a couple examples here. So this relies on those quantitative LCMS assays. And so we develop quantitative LCMS assays for these kind of series of compounds. In these ones, you don't need to actually do a full quantitation. It can be like a non-quantitative, and it can be like just change over time. So that's a lot better when you have like a whole series of things. This was actually a small panel. It's about 15 compounds. But if you were doing screening, you could do more than that. And so then we basically did these microsomal incubation assays to kind of understand their metabolism. We characterized the metabolism of these molecules basically by using LCMS to monitor them over time. As part of this, you can also monitor the production of metabolites. And so in this case, we were able to identify kind of a major metabolite and watch it grow in over time. And so metabolite ID can be part of your goals for studies like this if you want to understand what's the pathway of your drug's metabolism. We also look to see how the different structures influence the clearance of the drug and then calculated the half-lives. And so we use this similar, and we published this recently. So we use these kind of LCMS approaches to be able to quantify these novel drug compounds and understand some of their preclinical characteristics. So one last thing I wanted to say too, circling back to the proteomics. So say you have drug molecules and you're trying to understand their properties. And in this case, we worked with Steve Fletcher in our department where they had a CDK9 inhibitor. And one of their questions was what was the specificity of their molecule, and they had done a bunch of Western blots, and they'd actually gotten a request from a reviewer on doing a kind of a proteomics approach to see if they had any other off-target approach effects. And so we were able to take a proteomics effect and actually support their initial results to show that their molecule is actually very strongly only hitting the CDK9 and very minorly hitting a couple other ones and then nothing else. And so this proteomics approach can also answer questions like that. Um, so the last thing I want to just hit is we have some other kind of custom and unique capabilities in the center, including metal analysis, HDX, and oligonucleotide and other lipid structure function by other faculty members who also work in the center. So if you have things in that area, you might want to think about that. And then lastly, just recapping how we interact with us. Definitely do a consultation to talk about your experimental goals and scope and feasibility. Access those ICTR resources. Wanda, I'm sure, has posted the thing again. And then ultimately, we want to interact with you through grants and contracts. We're happy to help you with your grant preparation, letters of support. And ultimately, we work with people on funded projects and interact according to scope. It might be a small piece of your grant. It might be a major piece of your grant. That's why we like to talk with you to understand how best to interact and how best to accomplish your questions. And then I just shared, we have funding from a variety of sources over the years and a lot of great people who worked on these projects. And then I'm happy to answer any questions that you might have.
Maureen, that's amazing research, and thank you so much for providing such, wide scope of information and really in depth as well. I did place that link to the UMB ICTR website for drug discovery and development resources. That link is in the chat if you'd like to visit our website. And if anyone
Great.
has any questions, we can start taking those now. Anyone interested in utilizing these resources?
It was a lot of different stuff, so you might want to digest. Uh, I think what you said we're going to share the slides, Wanda, or
Yes.
they're available.
Yes.
So you can certainly think about this and also, make an appointment to chat about it.
I'm going to add the web page where we will be posting this recording, but I won't be there for probably a couple of days. And then also, if you'd like to raise your hand and just chat, you can use the raise hand feature and we can take your questions there. Okay. And okay. Going to Dr. Davis.
Oh, stop sharing.
I just placed the link in the chat as well to our education and training ICTR enrichment series webpage where this recording will be posted. And I believe Rachel will also be sending out a link to that recording to those who have attended for today. And if there's not any questions, we can give you back the rest of your lunch hour. And thank you again, everyone, for attending. And just a reminder, as you leave, you will be redirected to a survey. We really do appreciate any comments or input that you would like to add on future presentations. And again, thank you so much for attending. And we will give you back the rest of this hour. Everyone have a great rest of the day. Thank you, Maureen.